Okay, number 8. This is starting the new set of slides, virus to bacteriophages, and we will move on to bacteriophages in a little bit. But first of all, we're going to look at a few last things about animal viruses. We'll look at different kinds of infections we can get. We'll look at some other things that viruses can do to cells, and we'll look at how we grow cells. So, section 13.7, categories of animal virus infection. That's in the book on page 349. We can have acute infections. Rapid onset, short duration. So you get infected by influenza. The virus multiplies, and then you start to get symptoms. Your immune system kicks in. It eliminates the virus completely, and as soon as the virus levels are decreasing, those symptoms go away. In terms of influenza, you may later on get reinfected with another strain of influenza. In that case, you'll get another acute attack. But the main difference is that the influenza virus isn't persisting in you. Between attacks here and attacks here, you don't have influenza virus anywhere in your body. So that's what makes a persistent infection different, in that you get infected and the infection is never 100% cleared. The persistent infection may continue for months or years, or even an entire lifetime. You may have an infection, a viral infection, and you may not know it because you don't have any symptoms. These kinds of infections can be asymptomatic. Some viruses can have both acute and persistent phases, or indeed acute and persistent symptoms. HIV, when it first infects, it makes lots and lots of viral particles, and they cause this acute phase of infection, where you get fever and fatigue. HIV is a retrovirus, so what it does, it inserts its genome into your genome, and then it can't be snipped out, or it can't be removed by the human. And once it's integrated into the DNA, it is then persistent, and the virus can re-emerge. And you can then have this long period of time where you have the virus, and you can have a long state of infection. Okay, on to slide 3. If you have a persistent infection, it may be chronic or it may be latent. A chronic infection is an infection where levels of the virus particle are pretty constant. So here's our example of our chronic infection. Hepatitis B, the disease maintains a certain level, and you are then always getting symptoms. So the symptoms never really go away. The other kind of persistent infection you can have is a latent infection. And this is typically where symptoms come and go. So cold sores are a good example. You get an attack of cold sores and then they go away, and later on another attack and then they go away. But the difference being that in between times, the virus is still in your body. It's just typically hiding somewhere. When we come to latent infections, there are a couple of mechanisms that a virus can employ. it can hang around inside your cells, hiding as a provirus. And from the provirus, it can then reactivate. Or we can have some viruses like plasmids. Remember, plasmids from bacterial cells. We have our genome, and we have the viral genome as a separate element inside the cell. Once again, it can hide out, or it can be active. So a quick change there. The provirus cannot be eliminated from a cell. Once you have the DNA, the viral DNA, stuck into your genome, that segment of viral DNA is called a provirus, and it's there for good. Some viruses can move around the body. Herpes simplex 1, the virus that causes cold sores, can hide out in nerve cells called neurons, particularly in nerve cells that form these things called ganglia. Herpes simplex 1 does not integrate its DNA. It exists as a stable viral genome that hangs out inside the nucleus of infected cells. So lots of these latent infections their ability to cause disease is based upon the strength of your immune system. Kind of like a prisoner and a prison guard, or prisoners and prison guards. When there's lots of prison guards and all the prison guards are very active, then the prisoners behave. But as soon as the prison guards don't show up for work, or there's not enough prison guards, that's when the prisoners can break out of their cells and cause damage. So typically, when we have, for example, herpes, when it's hiding out in our ganglia, and a ganglia is a nerve junction where nerves from different parts of the body come together. For genital herpes, that ganglia is in the lower back. For the orofacial herpes that cause cold sores, the ganglia is located behind the cheekbones. And these latent infections can often be triggered by some kind of environmental condition. So when you are healthy and when your immune system is healthy, the virus is kept at bay. It's kept hiding. But as soon as your defenses begin to wane for any reason, including simple as stress. Stress has a very systemic effect on the body that can affect the immune system. Lack of sleep can cause stress. Hormonal changes, another disease. So you may get a cold, and then on top of the cold, you get a cold sore because your immune system is away fighting the cold virus, and the herpes virus then takes its opportunity to break out of its hiding place, break out of its cell, and then it too can start to cause problems. The virus then causes the symptoms of the cold sore, and that's how the virus can then move from one person to the next. You take a drink on a cup with a cold sore, the virus is now on the cup, and someone else mistakenly takes a drink from the same cup, and the virus has been passed from person to person. So different ways that viruses can affect us: acute or persistent, if persistent, chronic, or latent. Another important way that viruses can affect us is that they can cause tumors. Viruses in human tumors, 13.7 on page 351. So what is a tumor? A tumor is cells that have started to grow out of control. Abnormal growth. If a tumor has the ability to metastasize, it has the ability to spread from the original site of the tumor to other parts of the body, we would call that tumor cancerous or malignant. Benign tumors do not spread. So a normal healthy cell is being controlled by genes that make proteins. One kind of gene called a proto-oncogene. Those genes control the proteins that control and stimulate cell division. Another set of genes are tumor suppressor genes. Those are genes that control proteins, and those proteins inhibit cell division. So one way you can think about this is a car with a gas pedal and a brake pedal. And when the gas pedal and the brake pedal are both working, the car is safely traveling down the road. But if your brake pedal malfunctions and you can't put on the brakes, you have a problem. If your gas pedal gets stuck and you keep on revving the engine, then you have a problem. So whilst your gas and your brake are working, you're fine. In this case, cells do need to divide, so we do need that gas pedal. When you get a cut on your skin, we want to turn on cell division to fill in the gap. But we also want to make sure that we aren't making unhealthy cells. So we have to check and watch out for cells that are growing, and we need to inhibit cell division. So there's this balance. They work together to regulate growth and cell division. Mutations in the process of cell division can then interfere with these genes. A mutation in a proto-oncogene can change the behavior of the protein that gene encodes for, and the gas pedal is broken. Or a mutation in the tumor suppressor gene can, again, change the function of the protein that gene encodes for, and in that case the brake is broken. Usually the system isn't quite that delicate. You require multiple changes at multiple different sites in order to cause any kind of catastrophic car accident, if we're going to use that analogy. Proto-oncogenes and tumor suppressor genes, another example of names of things that aren't very helpful, because it sounds like a proto-oncogene is a gene involved in making tumors. But clearly there are no genes in your body that want to make tumors. These are cell types that were named because these are the obvious things that they did. This gene was identified because it was identified in a tumor, and it was seen that if you got a mutation or an interruption in this particular gene, then you could activate tumor growth. But these genes are regular genes that do regular stuff to keep regular cells being healthy. Proto-oncogenes can have many different functions in a cell. They might signal cell division. They might regulate programmed cell death. Tumor suppressor genes likewise have regular normal functions inside cells. typically to do with the cell cycle. So this is the cell cycle, a very simplified version of the cell cycle in mitosis. As the cell goes through the steps in mitosis, there are these kind of checkpoints, and these checkpoints are investigated by proteins, and these proteins are encoded in genes. So for example, the G2 checkpoint, this follows genome replication, and there is a protein that is able to take a look at your genome and see if there were any mistakes made. If there were mistakes made, then that protein may cause the cell to self-destruct instead of moving forward in the division process and potentially making unhealthy cells. Metaphase checkpoint are chromosomes attached to microtubules. When you pull the chromosomes apart, are you going to have any problems in pulling too many chromosomes in one direction and pulling too few chromosomes in the other direction? And the G1 checkpoint is cell division necessary. Has the body kind of signaled that cell division is required through growth factors, for example? There's no point going through the process of cell division if you're going to end up with a potentially harmful cell at the end. So what can cause the damage? How can viruses damage a cell and damage these genes to kind of turn them bad? Well, we've seen the idea of viruses inserting their DNA into the host DNA. And you can imagine then that we have our host DNA, and right here is a proto-oncogene. Here's our proto-oncogene on our chromosome. If we have the virus snip up our gene right there and insert the provirus, clearly the function of that gene has been destroyed, and say if that was an important gene in controlling the cell cycle, then the cell cycle may go bad. It may also be the case that as the provirus is coming in, and sometimes the provirus will snip itself out, sometimes it snips in the wrong place, and it can rearrange and it can affect the structure of genes and clearly have a significant effect. So if that occurs at the wrong place in your genome, then you can have troubles. If you have a chronic infection where a certain location is being continuously kind of bombarded with virus, and the immune system is trying to destroy the virus on a long-term basis, one of the outcomes of one of the outcomes of immune response is always inflammation, and inflammation, it's there to get rid of the invading pathogen, but it's also very damaging to our own cells. And that damage can then be seen in damage to the DNA. Damage to the DNA in the wrong spot leads to a dangerous cell that is replicating out of control. And then number 3, the virus may contain on it genes. So once again we have our provirus stuck into our genome, and this virus itself has genes. One of these genes may be turned into a protein, and then that protein has some kind of effect on cell division. The viral gene in the provirus interfering directly with some process in cell division. And this, if you're looking from the virus's point of view, this is good for the virus because more cells interrupt the cell cycle, cause a cell to multiply out of control. More cells means more targets for the virus to infect, and that means more virus. And that's the entire point of a virus, is to make more of itself. These viruses that can interfere, that carry with them oncogenes, are called oncogenic viruses. They're typically DNA viruses, and HPV is probably an example that we have all heard of in regards to a virus that can cause tumors. The last thing I want to mention about animal viruses is cultivating them. Animal viruses and viruses in general can only live and only be cultivated in growing cells. And used to be done by inoculating live animals with the virus. And the virus multiplies inside the animal, and later on we can then extract the virus. Why do we want to grow viruses? We want to grow viruses because we want to study them, is one important feature. We moved on to fertilized chicken eggs. We now use cell cultures, and tissue cultures are more common. There's a little diagram on page 353 about producing primary cultures. The downside of many tissue cultures or cell cultures is that the cells won't divide beyond a certain point. One of the reasons why we haven't been able to, say, grow replacement organs is it's very hard to get the cells to keep on growing in this kind of artificial environment. One thing that we can then use for viruses is tumors themselves. Cells that have been damaged in such a way that they are replicating out of control. They can multiply indefinitely, and therefore they can maintain a virus population also indefinitely. I will fill in the study guide, but we've talked about acute versus persistent, persistent being chronic or latent, so be able to identify the type of infection from a description, know how some of these viruses, some of these latent viruses can, where they hide and how they are able to hide, know how viruses can cause tumors, know what a proto-oncogene and a tumor suppressor gene, know what they do inside cells, and know that we must have living cells for viruses to grow. Okay, that's that.